A very good morning to you all. In this section, we are going to learn about chromosome and its structure. These are the things we are going to learn in this section. First, we should know about chromosomes. Chromosomes are thread-like structure present in the nucleus, which is carry genetic information from one generation to another. They play a vital role in cell division, hereditary, variation, mutation, and also repair and regeneration. Each chromosome is made up of tightly coiled DNA around protein called histone that support its structure. The chromosomes means color body that refer to its staining ability by certain dyes. The term chromosome was first coined by W. Voltaire in 1888. The number of chromosomes in any species is constant for all these cells. The chromosome numbers varies in different species. For example, the nematode species contain only 2 chromosomes in all cells, whereas a protozoan species contain as much as 1600 chromosomes in the cell. A human cell contains totally 23 pair of chromosomes, of which 22 are autosome and 1 sex chromosome. The size of chromosome ranges from zero point 0.1 micron to 30 micron. The diameter varies from 0.2 micron to 2 micron. The length of human chromosome varies from 4 micron to 6 micron. But the giant chromosome varies in length and diameter. For example, Lamprecht chromosome in oocyte of amphibian may reaches up to 800 microns. Then the karyotyping is a technique to study structure of chromosome present in a species. This is the structure of chromosome. The main parts of chromosomes are chromatide, chromonimeta, chromomeres, centromere, secondary construction or nucleolar organizer, telomere, and satellite. The first part of the chromosome is chromatide. Each chromosome has two symmetrical structure called chromatide or the sister chromatides, which is visible in mitotic metaphase. The next one is chromonimeta. Internally, chromosome contain two identical spirally coiled filaments called chromonimeta. At prophase, the chromonimeta is visible as thin filaments. The nature and degree of coiling of chromonimeta is variable in mitotic and meiotic chromosome. In meiotic chromosomes, two distinct coils are observed. One is the major coil consists of 30 to 10 to 30 gears. Other is a minor coil has more number of gears. In the meiotic chromosomes, is similar to the mature coil of somatic or standard coil founded. The coiling may be either paranemic, where the coil can be easily separated, or pleconemic, where the coiling cannot be easily separated. At interphase, chromosomes are visible as thin chromatin fiber present in the nucleoplasm. During the cell division, the chromatin fibers condense and chromosomes are visible with distinct feature with a filigrane stain. The darkly stained condensed region of chromatin is known as heterochromatin. It is contained tightly packed DNA which is genetically inactive. The lightly stained diffused region of chromatin is known as euchromatin. It contains genetically active and loosely packed DNA. This phenomena is known as heterophycnosis or differential staining. The next part of chromosome is chromomere. At interphase, the bead-like structure are visible in the chromosome, which are accumulation of chromatin material called chromomere. Chromatin with the chromosomes look like a necklace with the beads. The next one is centromere and kinetochore. Sister chromatin are joined by the centromere. The number of position of centromere differs in the different chromosome. The centromere is called primary construction. Centromere divides the chromosome into two sections or arms. The shorter arm is called P arm and the longer arm is called Q arm. The centromere has 5 zones, namely inner zone, 2 middle zone and 2 outer zone. The inner zone of centromere contain a disc-shaped kinetochore, which has specific DNA sequence with a special protein bound to them. The next part is secondary construction or nuclear organizer. Constant in their position and extent, the construction are useful in identify particular chromosome and a set. Secondary construction can be identified from centromere at anaphase because they are binding only at the centromere. Secondary construction, which contains gene to form nucleoli, or known as the nucleolar organizer. The next part is telomere. The terminal part of a chromosome is known as telomere. These are specialized end of chromosome which exhibits a physiological differentiation and polarity. Telomere are polar, which prevent the fusion of the chromosome segments. The next one is satellite. The chromosome region distal to the secondary construction, for example, the region between the secondary construction and the nearest telomere is known as satellite. It is an elongated segment that sometimes present in a chromosome at the secondary construction. The chromosome which satellite are known as sat chromosome. So these are the things about the chromosome and its structure. I think this section may be very useful to you all. Thank you.